Well, good morning. It's really an honor and pleasure to speak about microglia, a very different topic this morning. So, as the provocative title of this research article suggests, we are experiencing an important paradigm shift in the microglia field. Microglia, which are the immune cells of the brain, are not toxic cells that cause inflammation and that should be eradicated. Here, inflammation refers to the defensive activity of the immune system towards pathogens and other challenges that we can experience. Instead, microglia are being shown to be beneficial cells that actively contribute through their many physiological roles to maintaining brain, but also behavioral and cognitive health along the aging trajectory. From this excellent article here I'm reading, although microglial activation is often considered neurotoxic, microglia are essential defenders against many neurodegenerative diseases. It also seems increasingly likely that microglial dysfunction can underlie certain neurological diseases without an obvious immune component. So this is what I'm going to speak about, the role of microglia in brain diseases along the aging trajectory, giving an overview of the current knowledge in the field. Over the past decade, the beneficial roles of microglia in the healthy brain have started to unravel. What you can see here is a search online with PubMed using the term microglia in all the fields or in the title of the articles only. There were only few research papers published on the topic of microglia before the 1990s, and then there's a steady increase of this research on microglia until the beginning of our century, leading to an exponential growth of microglial research. You can notice on this graph a first inflection point in 2005 or 25, which coincides with really an important discovery in the field using non-invasive imaging, a discovery showing that microglia, which used to be called resting or quiescent in the healthy brain, are actually extremely dynamic. Microglia continuously survey the brain with their highly motile processes. There's also another inflection point in 2010, which coincides with the discovery of the exclusive origin of microglia from the embryonic yolk sac. The yolk sac is a structure that nourishes the developing embryo. Microglia come from this embryonic yolk sac. They enter the brain during development, and then they stay there throughout life. And also in 2010, my work as a postdoctoral trainee revealed that microglia surveying the brain with their highly motile processes frequently interact with synapses. Synapses are the connections between the neurons in the brain, and this takes place in health during normal physiological conditions and contributes to removing or pruning, as illustrated in this cartoon, the less active connections between the neurons in the brain. Whenever the homeostasis of the brain is altered due to trauma, injury, or infection, microglia rapidly transform their structure and their function on a timescale of minutes. You can see at the right this transformation of microglia from ramified cells to cells looking more like amoeboid macrophages as they increase their ability to release toxic mediators, inflammatory mediators, also as they increase their ability to phagocytose or remove pathogens, degenerating cells, different types of debris in the brain. By contrast, the roles of quiescent or resting microglia in the healthy brain, which are illustrated at the left, had remained unknown until very recently, because microglia transform with the same rapidity in response to experimental procedures used to study microglia. You can see at the left, as I mentioned, these quiescent or resting microglia. These are drawings from Pio del Rio Ortega, who is considered the father of microglial biology, drawings from 1919. With the development of non-invasive imaging methods, the previous findings in neuroscience, mainly from cell culture or other invasive experimental procedures, for instance, brain slices, that suggested that microglia are toxic cells that should be eradicated, have been questioned. Here I will read from this excellent article in the field. From an evolutionary point of view, it is difficult to understand why an important, mostly post-mitotic and highly vulnerable organ like the brain would host numerous potential killers. This review aims to critically reconsider the term microglial neurotoxicity and discuss experimental problems around microglial biology that often have led to the conclusion that microglia are neurotoxic cells. Here I'm illustrating this rapid response of microglia to injury. It's a video which was captured using non-invasive imaging in the brain of a mouse. The microglia are in green, so I'm starting the video. You can see these dynamic cells that respond to the damage to the neurons. The damaged neurons are in red. Microglia extend their processes and transform into these amoeboid phagocytes, similar to macrophages that eliminate pathogens. But this is in the brain in response to injury. The development of these non-invasive imaging methods was necessary for the understanding of the roles of microglia in the healthy brain. And this has led to a subsequent exponential growth of research in the field. Here I'm showing pioneer findings from 2005, showing that quiescent or resting microglia observed for the first time in the intact brain of living mice are extremely dynamic. This was very surprising to the field and has really contributed to shaping the research. You can see in the overlay at the right, the difference in morphology between two images of the same microglia that were captured 60 minutes apart. All the multiple processes surveying the brain are shown in green or red, and the cell body of the microglia, which is stable, keeping its position in the brain, is shown in yellow with the merge of the green and the red fluorescence. Using non-invasive imaging in my postdoctoral work, I was also able to show that microglia make dynamic interactions with synapses, the connections between neurons. Neurons are the main cells of the brain. They communicate one with another, and they remodel their connections to allow for learning, memory, also the adaptation to the environment. In this video, the microglia is in yellow. You can notice that the cell body, this is in a living mouse, is relatively stable in this corner of the video, but the processes are extremely motile and contacting these synapses shown in green. As I mentioned, the neuronal circuits in the brain are constantly remodeled. This takes place through the formation, the modification, or the elimination of the synapses, these connections between neurons. And this is what allows for learning and memory and the adaptation to the ongoing changes in the living environment. Based on non-invasive imaging observations, notably from my postdoctoral work, using electron microscopy or high-resolution technique, there's different modes of microglia-mediated synaptic elimination that were identified. For instance, there is this phagocytosis, which I mentioned before, that can be directed towards pathogen or damaged cells to remove them from the brain, but that can also serve to eliminate these connections between the neurons, to change the pattern of connection of the neuronal circuitry. This is what is illustrated here. And there's another mechanism that we study a lot in the lab. It's called synaptic stripping. This refers to the physical separation of the pre and the postsynaptic elements by intervening microglial processes, which physically interrupt the transmission between the neurons. At the right, I'm showing with this electron microscopy an example of phagocytosis. This is a microglial cell, and there is this phagocytic inclusion. It's a presynaptic terminal containing vesicles with neurotransmitters, the chemical messengers used by neurons to communicate in the brain. And this is found inside of the microglia in the process of being eliminated, of being digested with enzymes. With these findings and several other studies in the field undergoing an exponential growth, microglia are now considered to be crucial during development for the survival of the neurons, also for the clearance of the dying cells, the formation of the synaptic connections between neurons, also for the elimination or pruning of less functional synapses. In adulthood, microglia are also important effectors of brain plasticity, contributing to this neuronal circuit maintenance and remodeling, learning and memory, also the adaptation to various environmental challenges that we can encounter during life, such as chronic stress. And then over the course of aging, but also in diseases, microglia become more diverse, and they can also become altered or compromised in all of these beneficial physiological functions, leading to the emergence of various disorders and diseases throughout life. When microglial function is compromised, there's a lot of consequences along the aging trajectory. These beneficial immune cells are quite important. This is what the new research is showing. In different studies using animal models of neuropsychiatric disorders or neurodegenerative diseases, rodents, also non-human primates, treating microglial dysfunction, normalizing their beneficial functions, as revealed overall, depending on the stage of life, improved outcomes on cognition, also social behavior, learning-dependent synapse formation, locomotion, anxiety, risk assessment, as well as neurodegenerative disease pathology, spatial memory, and male sex behavior, for instance, in adulthood as well. And then in human treatment with the antibiotic minocycline, which is prescribed for the treatment of acne, was shown to dampen inflammatory responses and also to normalize microglial functions. For instance, their phagocytosis that they can use to eliminate a lot of elements in the brain, including synapses. And this was shown to reduce the rewarding effects of methamphetamine abuse, producing beneficial outcomes in major depressive disorder when added to treatment with selective serotonin reuptake inhibitors, and also to reduce positive and negative symptoms in early phase of schizophrenia when added to conventional antipsychotic treatment. Recently, cutting-edge molecular techniques have revealed that in the healthy brain, during normal physiological conditions, microglia are highly diverse during development, and then they mature to adopt this surveillance state that I have shown before, a state that is conserved across species, across rodents, non-human and human primates as well. But then they diversify again over the course of aging based on the various influences from the environment that they receive, to which different microglial cells are differently exposed across life. And these findings suggest that the multiple physiological roles of microglia that are exerted in the healthy brain, notably during development, could rely on distinct specialized subpopulations of microglia. The changes of microglia during the lifespan also differ between males and females. For instance, in mice, male microglia were found to be bigger and to have a lot of differences also on the molecular level. for instance, an increased production of pro-inflammatory mediators. While microglial sex difference remains still largely unknown, they were shown to be driven by a combination of gonadal hormones and also sex chromosomes. Since Pio del Rio Ortega, the father of microglial biology, first described a first subpopulation of microglia named the satellite microglia, a subset that is associated with the neurons, these principal cells of the brain, and regulate the brain activity. There's a large number of microglial subpopulations that were discovered. It's something that is quite emerging in the field now, this notion that microglia are very diverse, very heterogeneous. And among these different populations, there's the dark microglia that my group has described in 2016. These dark microglia were described using non-invasive imaging techniques, particularly electron microscopy, using their unique markers of cellular stress and metabolic alteration. These cells are very abundant during early development, but then as the animals mature, they become very rare. There's very few of these cells in healthy adult mice. And they become, again, very prevalent with the exposure to chronic stress, also aging, in different models of neurodevelopmental and neuropsychiatric disorders and neurodegenerative diseases, suggesting a strong link with pathology. These cells display exacerbated interactions with synapses, these connections between the neurons, phagocytic and non-phagocytic interactions, suggesting their implication in the pathological remodeling of neuronal circuits, which was shown to lead to defects of learning and memory, also cognitive impairment during aging. These dark microglia have now been documented by my groups and others in various animal models, as well as in human. And they appear as a quite promising target for specialized interventions, normalizing these altered functions of dark microglia. Before concluding, I wanted to mention some perspectives on microglial regulation by various environmental influences, including risk factors for disease. The exposure to environmental risk factors such as chronic stress, combined with the individual genetic predispositions, results in the onset of different neuropsychiatric disorders and neurodegenerative diseases, depending on the stage of life, and depending also on the sex of the individual. Development is a particularly sensitive period. Prenatal stress and also infections, notably with viruses, combined with other challenges during life, such as dietary imbalance, pollution, sleep disturbance, over a lifetime are important risk factors for the development of autism spectrum disorders, but also schizophrenia, major depression, other mental illnesses. And then with aging, they are important risk factors for the late onset or sporadic forms of Alzheimer's disease, Parkinson's disease as well, other similar causes of dementia. A large number of developmental and adult psychiatric disorders and age-related neurodegenerative diseases are similarly associated with a combination of environmental risk factors that compromise the beneficial functions of microglia differently between the sexes and depending on the stage of life, on top of the genetic risk factors that confer a vulnerability. So overall, these findings propose a paradigm shift in the microglia field from a unique cell type, microglia, that respond to environmental challenges, but also the diet and different lifestyle factors, as well as disease pathology, by becoming neurotoxic, activated, or reactive across the lifespan, to a community of diverse cells, of heterogeneous microglial cell populations that each perform different physiological functions, respond differently to environmental challenges, lifestyle factors, also to disease pathology. depending on the sex of the individual and the different environmental exposures throughout life. Our research on the dark microglia and other subsets aim to provide novel therapeutic avenues for a wide range of neurodevelopmental and neuropsychiatric disorders, also neurodegenerative diseases in which microglial dysfunction is implicated along the aging trajectory. I am grateful to my team, collaborators, also funding sources for the essential support. Thank you for your attention. I will be happy to take your questions.